Hello, welcome to this video again, and we will continue from where we ended last and see what we have here. We have some of the steps in the production of monoclonal antibodies. Okay, a mouse injected with antigen A. Mouse is led for a few days to allow immune response to occur. Plasma cells are extracted from the mouse. Hybridoma cells are formed. Each hybridoma cell is isolated and allowed to grow and divide. The hybridoma cells producing antibody A and identified, cultured on a large scale. Okay, so with reference to figure 2.1, explain what happens during an immune response. Okay, with the reference to figure 2.1, during an immune response, plasma, okay, maybe we have to give the details of the things that happen in immune response. Okay, that's AS part, but then we can just recall. So during an immune response, the antibodies, actually the lymphocytes produces. So first thing we should know that lymphocytes produces, this one produces, okay, this is the first point, important point. But then if you want to go in the sequence, let us just see what do they have in our marking scheme. Antigen presenting cells, okay, antigen equals 9 as non-self by B lymphocytes with appropriate receptor, clonal selection, B lymphocyte clonal expansion, mitosis, cell division, T helper cells to stimulate B cells, release cytokine, matures to B lymphocyte mature into plasma cell, secretes antibody. Okay, so you have to go with this, with this sequence that there is this the cells, okay, antigen presenting cells. We make this antigen go inside, right? We make this antigen go inside. Okay, sorry, no. We make this antigen go inside. Antigen and recognize a non-self. So the first thing it goes. The first thing. It is the first thing. This is the first thing. Second thing is given, is recognized and non-self. Okay, that is the second thing. Then the third thing. This is done by B lymphocyte, okay, B lymphocytes. B lymphocyte, okay? And then the third thing is, should also say that how this is possible is because of wait, let me just move this here so I can finish my sentence maybe. Okay, let me just keep this here, and let me just keep this B lymphocyte, okay, B lymphocyte, and then with appropriate receptor, appropriate receptor. So it is identified, the first part is identified, and then the second thing is this is goes in, okay, this is goes in, goes in, okay, and then it is identified as non-self by B lymphocyte by the appropriate receptor, and then after that B lymphocyte actually what it does is there is a clonal selection, means B lymphocyte reproduce, okay, and then after clonal selection the clone that is supposed to be produced is, so the first thing happens is clonal selection, as we said here. Now you should understand that when we have a lot of antibodies, I mean in the body we have a lot of B cells, right? So when antibody, the first thing is it should be identified as non-self. Okay, the cell which comes in should be identified as non-self. Okay, that's the first thing, and then there should be clonal selection. It should be identified that which cell, I mean what antibody, what receptor does the antigen has, so we can produce a complementary antibody. Okay, so clonal selection is simply the identification of the receptor, this receptor, okay, and the consequent production after clonal selection. That's why we get clonal expansion. Okay, we get clonal expansion after clonal selection. We got clonal expansion, right? So hope I made my point clear. And then T helper cells stimulate B cell. Okay, T helper cells stimulate B cells. Yeah, this is also known that, okay, actually after the clonal expansion, so this process is actually this process is the one actually there are this another cells, T helper cells, which stimulates B cells to release cytokines. Okay, B lymphocyte mature into plasma cells, then these clones, they mature into plasma cells, and these are the cells we want. Okay, so hope I made that point very clear. And these plasma cells secrete antibodies from these plasma cells. Okay, I guess I'm clear with what I just said, hopefully. This is another thing, I mean it's not regarding this one. This is T helper cells stimulate B cells to release cytokines. Okay, these cytokines are the one that comes and influence this whole process. Okay, so hope you got this concept over here. This is just a revision actually. This is an AS part. Okay, the immune response. You can go and refer to the books for more proper steps. And I think with that I will conclude that I finished this part. I'll continue the next question in the next part.